Hello guys, this is Jyoti Rana. Hope you all are doing well. Today I'm going to discuss the last part of this chapter, cell, the unit of life. So let's start with the ribosome. Ribosomes. We had already discussed about the ribosomes. Ribosomes are the granular structures first observed under the microscope as dense particle by the George Palade in 1953. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. Ribosomes are non-membrane bound structure. The eukaryotic ribosomes are 80S, while prokaryotic ribosomes are 70S. Each ribosome has two subunits. This is the large subunit, this is the small subunit. And the two subunits of 80S ribosome are 60 and 40. 80S ribosomes, 60S plus 40S. 60S is the large subunit, 40S is the smaller subunit, while that of 70S ribosome, 50S plus 30S. For the 70S, it is 50S plus 30S. S is the Swedberg unit, stand for the sedimentation coefficient. It is indirectly measure the density and the size. Both 70S and 80S ribosomes are composed of two subunits. We know that mitochondria have 70S, mitochondria and chloroplast 70S ribosomes. But these are organelles present in eukaryotic cells. So, in the eukaryotic cell, 80S ribosome present in the cytoplasm and 70S ribosome present in the mitochondria and the chloroplast. The next is cytoskeleton. Cytoskeleton. Cyto means cell. Skeleton means अपना ढांचा. So, यहाँ पे हमें तीन टर्म्स पता होनी चाहिए. Exoskeleton, लाइक इन कॉकरोच की जो हार्ड कवरिंग है वो उसका स्केलेटन है. सो वो बाहर है तो वो एक्सोस्केलेटन. हमारी जो हार्ड बोन्स होती हैं वो अंदर है, सो हम उसको बोलेंगे एंडोस्केलेटन. अब स्केलेटन क्या कर रहा है? मेंटेन कर रहा है किसी चीज को, बॉडी को मेंटेन कर रहा है, उसकी शेप को मेंटेन कर रहा है, मैकेनिकल सपोर्ट कर रहा है. सो साइटोस्केलेटन मीन्स सेल में के कुछ स्ट्रक्चर्स, सेल में कुछ स्ट्रक्चर्स प्रेजेंट होते हैं, जैसे हमारे अंदर हमारी बोन्स है तो उनको हम सेल साइटोस्केलेटन बोलेंगे. एन एलाबोरेट स्ट्रक्चर ऑफ फिलामेंटस प्रोटीनियस स्ट्रक्चर. ये प्रोटीनियस स्ट्रक्चर होते हैं consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm is collectively referred as cytoskeleton. These fibers, these proteins are like the fibers and tubules. Tubes की तरह होते हैं, fibers की तरह होते हैं. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. ये इनके फंक्शन है. मैकेनिकल सपोर्ट करके रखता है, सेल को मेंटेन करता है, उसकी शेप को मेंटेन करता है, एंड द वन मेजर फंक्शन इज मोटेलिटी. अब मोटेलिटी यूकैरियोटिक सेल्स में तो मोटेलिटी है नहीं, बट एक सेल है यूकैरियोटिक ऑर्गेनिजम्स में, ह्यूमन में मोटाइल सेल. Which one is that? That is sperm. Human sperm. This is the flagella. And this flagella is made up of microtubules. सो ये क्या कर रहा है, मोटिलिटी में हेल्प कर रहा है. फ्लैजला इज अ स्ट्रक्चर ऑफ माइक्रोट्यूब्यूल्स. माइक्रोट्यूब्यूल्स का है, these are the microtubules. Microtubules is a type of cytoskeleton. अब कर लेते हैं. Cytoskeleton, a proteinous structure like fibers and tubules consisting of microtubules, intermediate filament, and the last one is microfilaments. Microtubules ये तीनों सेम होते हैं, but the difference is their diameter. Microtubules are like this. Intermediate filament, इनका diameter थोड़ा कम होता है, ट्यूब्स की तरह ही है. माइक्रोफिलामेंट और छोटा डायमीटर होता है, like this. These are made up of ट्यूबुलिन प्रोटीन. ट्यूबुलिन प्रोटीन अभी हमने देखा था फ्लैजला का एग्जांपल. फ्लैजला में ट्यूबुलिन प्रोटीन माइक्रोट्यूब्यूल्स होते हैं जो कि ट्यूबुलिन प्रोटीन के बने होते हैं. सो फॉर एग्जांपल cilia and flagella and centriole. These are also made by the microtubules. Intermediate filaments, keratin proteins like हमारे हेयर में होते हैं केराटिन प्रोटीन, हेयर लाइक स्ट्रक्चर, थिन होते हैं, माइक्रोट्यूब्यूल्स से थिन होते हैं. माइक्रोफिलामेंट, एक्टिन एंड मायोसिन प्रोटीन. एक्टिन एंड मायोसिन कहाँ होते हैं, इन मसल्स में. Here, that's it. Cilia and flagella. Come to the blank slide. Firstly, we discussed the structure of the cilia and flagella. Cilia and flagella the cilia in the high number में प्रेजेंट होता है, फ्लैजला लेस नंबर में प्रेजेंट होता है. सीलिया छोटू होता है, फ्लैजला लंबा होता है. सो द स्ट्रक्चर इज सेम. सीलिया एंड फ्लैजला का जो स्ट्रक्चर है वो सेम होगा, लाइक दिस. 1 2 1 2. Sorry, I should rub this. Centrosome is an organelle usually containing two cylindrical structures called the centriole. Come to the blank slide. This is a cell. During the division, chromosomes are like this. In the metaphase, chromosomes are like this. Here is the centrosome. Two perpendicular centrioles on the both side of the cell. Okay. Some This is the part of pericentriole. Centrioles are nothing else. These are the microtubules. Microtubules. जैसे cilia and flagella का स्ट्रक्चर था ऐसे ही इनका भी. But the difference is the formula is 9 plus 0. It means there is 9 triplets, not doublets. 9 triplets like this. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3. 1, 2, 3. 9 triplets are there. They are joined with first to third, first to third, first to third, like this. This is the structure of centrioles, and the area between this is known as hub. So 9 triplets are present in the centriole, and सेंटर में एक हब लाइक मतलब प्रोटीन होते हैं, स्ट्रक्चर कुछ नहीं होता, कोई माइक्रोट्यूब्यूल प्रेजेंट नहीं होता सेंट्रियोल में. सो 9 plus 0 is the formula of centriole. This is the structure of metaphase stage. हम सेल डिवीजन में पढ़ेंगे, स्टेजेस है, इंटरफेज, देन प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज. Like this, in this chromosome, this is the centromere. And a disc-like structure is present near the centromere that is known as kinetochore. In the metaphase, spindle fibers are formed. Spindle fibers, वो क्या होंगे? ऐसे. They are attached with the chromosomes. These are the spindle fibers. These spindle fibers are also microtubules. In the anaphase stage, this is the stage of anaphase during the cell division. The spindle, these are the centrioles. The spindle fibers detach the chromosomes. So this is the stage of anaphase. सेल डिवीजन में पढ़ेंगे. स्पेशली यहाँ हमें क्या याद रखना है? सेंट्रियोल made up of microtubules. Spindle fibers made by the microtubules. And the chromosome, this is the centromere. This is the kinetochore, where the spindle fibers are attached. Centrosome is an organelle usually containing two cylindrical structures called the centrioles. They are surrounded by amorphous pericentriolar material. Pericentriolar material, This is the pericentriolar material. Both the centriole in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. Cartwheel किधर है? This is the structure of the centriole. The nine triplets are present, A, B, and C. A attached with the C. These are microtubules. These are the spokes, and this area is the hub, central hub, pinhead, AC linker. Each of the microtubules are like this. These are the centrioles. This is the pericentriolar material, proteinaceous, and this structure is not bound with any membrane, not bound with any membrane. They are made up of 9 evenly spaced peripheral fibrils of the tubulin protein. We know that microtubules are made up of tubulin proteins. Each of the peripheral fibril is a triplet. Adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinous and called the hub, which is connected with the tubules of the peripheral triplets by the radial spokes made up of protein. The centriole forms the basal body of the cilia and flagella and spindle fibers that give rise to spindle apparatus during the cell division. Spindle fibers हम already पढ़ लिया है हमने. Spindle fibers सेंट्रियोल के बने हुए होते हैं. अब what is the meaning of basal body of cilia? This is the flagella, and the basal body of this flagella is this. The basal body is made by the centriole. So the arrangement is like 9 plus 0, and the arrangement of flagella is 9 plus 2. So this is the meaning of basal body of cilia and flagella. And spindle fibers we had already discussed. Then come to the nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Nucleus stained by the basic dyes was given the name chromatin by the Fleming. This is the structure of the nucleus. This is the outer membrane, and this is the inner membrane. Nucleus have two membranes, outer membrane and inner membrane. The space between the outer membrane and inner membrane is perinuclear space. Perinuclear space. This is the nucleolus. Some fluid area in the nucleus, dense area in the nucleus is nucleolus, and these are the nuclear pores. These nuclear pores are help in the transportation of RNA and protein. सब प्रोटीन्स अंदर आएंगे, आरएनए बाहर जाएगा, तो उनको आने जाने के लिए जो रास्ता चाहिए वो कहाँ से होगा? थ्रू दी पोर्स. न्यूक्लियर मेम्ब्रेन हो गया. दिस इज न्यूक्लियोप्लाज्म. The fluid in the nucleus is known as nucleoplasm, like the cytoplasm. सेल के अंदर का जो फ्लूडी एरिया है that is known as cytoplasm, and nucleus के अंदर का जो फ्लूडी एरिया है that is nucleoplasm. And this is the chromatin. Chromatin, we know that chromatin is a DNA, is a genetic material, is a chromosome. So ये हम डिस्कस करते हैं. Come to the blank slide. What is DNA? DNA is a, this is DNA, diribonucleic acid, DNA. Okay. When the proteins, some histone protein होंगे, when the proteins enter into the nucleus and they bound with the DNA, these are the histone proteins. Histone protein. These histone proteins are bound with the DNA like this. Like this. And they form nucleosome. This is this histone protein plus DNA is known as nucleosome. The structure of histone protein and DNA is known as nucleosome, and they form a condensed structure like this. This is the chromatin. है सब कुछ डीएनए. जब डीएनए सिर्फ अकेला डीएनए है तो वो डीएनए है. देन when DNA bound with the histone protein and plus DNA, the unit, a single unit is known as nucleosome. When histone protein wrap with the DNA wrap into the histone protein, they form chromatins. ये क्यों करना पड़ा? जो हमारा डीएनए है उसकी लेंथ 2.2 मीटर है इन अ सेल. सेल कितना छोटा है, सेल में न्यूक्लियस कितना छोटा है, वहां पे इतना बड़ा डीएनए कैसे एडजस्ट होगा? सो उसके लिए क्या होता है, पैकेजिंग होती है डीएनए की. ये पैक हो रहा है, यहाँ पे पैकेजिंग हुई डीएनए की, क्रोमेटिन बना. अब ये क्रोमेटिन क्रोमोसोम बनाएगा. कंडेंस्ड फॉर्म ऑफ डीएनए, कंडेंस्ड क्रोमेटिन, कंडेंस्ड. सो ये क्रोमेटिन है. जब वो लूज, थोड़ा लूज है, तो लूज जैसे इंटरफेस में ऐसे ही पड़ा होता है डीएनए इन द न्यूक्लियस. दिस इज क्रोमेटिन. अब मेटाफेज में क्या होगा, ये क्रोमोसोम बन जाएगा लाइक दिस. सो इसी डीएनए को हम यहाँ पे क्रोमोसोम बोलेंगे. सो द क्रोमोसोम इज द कॉम्बिनेशन ऑफ डीएनए एंड प्रोटीन. This is all about DNA packaging. Okay. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both the directions between the nucleus and the cytoplasm. So transport of RNA and protein takes place through the nuclear pores. Normally there is only one nucleus per cell. Variation in the number of nuclei also frequently observed. Some mature cells even lack nucleus. This is the very important data. प्रीवियस ईयर क्वेश्चंस भी हैं. कौन सी सेल्स में न्यूक्लियस नहीं होता? मैमल्स में क्या है, एरिथ्रोसाइट्स ऑल्सो नोन एज आरबीसीज, एंड सीव ट्यूब सेल्स इन द वेस्कुलर प्लांट्स. उनमें भी न्यूक्लियस नहीं होता बट फिर भी वे लिविंग है. एंड द नेक्स्ट इज न्यूक्लियोप्लाज्म. The nuclear matrix or the nucleoplasm contain nucleolus and the chromatin. This is the structure of nucleus, chromatin, nucleolus, all known as nucleoplasm. The nucleoli are spherical structure present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. This just the content, dense content, nucleolus, nucleolus, and not a membrane bound structure. ये नॉन मेम्ब्रेन बाउंड स्ट्रक्चर है. सो इट इज कंटिन्यूअस विद द रेस्ट ऑफ द न्यूक्लियोप्लाज्म. इट इज द एक्टिव, इट इज अ साइट फॉर एक्टिव राइबोसोमल आरएनए सिंथेसिस. अब डीएनए यहाँ हो रहा है, डीएनए यहाँ बन रहा है, क्रोमोसोम डिवीजन यहाँ हो रही है, तो आरएनए भी यही बनेगा. दिस इज द आरएनए. Larger and more numerous nucleoli are present in the cells actively carrying out protein synthesis. So RNA बना है तो protein synthesis भी होगा. The interphase nucleus has a loose, indistinct network of nucleoprotein fibers called the chromatin. This is the loose structure of the DNA that is known as chromatin. But during different stages of cell division, cells show structured chromosome in the place of nucleus. Chromosomes condensed form में convert हो जाएगा DNA. Chromatin contains DNA and some basic proteins called the histone proteins. Some non-histone proteins and also RNA. Chromatin में ये भी present होते हैं. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 chromosomes. कितने chromosomes होते हैं human body में? 46 chromosomes. It means 23 pairs of chromosomes. Every chromosome, visible only in dividing cells, essentially has a primary construction or the centromere on the side of which disc-shaped structure kinetochores are present. अभी थोड़ी देर पहले हमने देखा था, this is the centromere and the disc-shaped structure is known as kinetochore. Centromere holds two chromatids of the chromosome. These are the one chromatid and this is the second chromatid. This is the one chromatid and this is the second chromatid. Types of chromosome based on the position of the centromere. The first one is metacentric. Metacentric. In this chromosome, centromere present in middle of the chromatids and form the equal arms, two equal arms. This type of structure is known as metacentric chromosome. And the satellite is also present. It is a secondary construction of satellite of chromosome is known as satellite. It is present in few chromosomes. Few chromosomes. Then come to the second one, submetacentric chromosome. In this chromosome, centromere slightly away from the middle, so they form the one short arm and one long arm. Then acrocentric chromosome. In the acrocentric chromosome, centromere near the end of the chromosome, it forms extremely short arm and a extremely long arm. Then last one is telocentric. In the telocentric, centromere situated at the end of the one arm. So there is only one arm. Second arm is very extremely short. We can say there is only one arm. So this is the kinetochore, disc-like structure. Metacentric chromosome has middle centromere forming two equal arms. Submetacentric chromosome has centromere slightly away from the middle, so resulting one shorter arm and one longer arm. In case of acrocentric chromosome, centromere situated close to the end forming one extremely short and one very long arm. Telocentric chromosome has a terminal centromere. बिल्कुल एंड पे होता है, सिर्फ one type of arm ही present होती है. Sometimes a few chromosomes have non-staining secondary constructions as a constant location. This gives the appearance of a small fragment called the satellite. So, this is the chapter cell, the unit of life, are about to finish. So, the next chapter is the cell cycle. So, if you like my videos, do like, subscribe, and share my channel. Thank you so much.